Today we have the pleasure of hosting Dr. Charlotte Cunningham-Rundles, Dr. Yaron and Dr. Ioannis Tassiulas for a multidisciplinary review on autoimmunity. This unique grand rounds will allow for a brief overview of current research in the divisions of allergy and immunology, endocrinology, and rheumatology, and highlight a disease across multiple disciplines. Dr. Cunningham-Rundles is the David S. Gottesman Professor of Immunology at the Mount Sinai School of Medicine. She's a professor of medicine and pediatrics, a member of the Immunology Institute, and directs the Immunodeficiency Clinic at Mount Sinai. She received her MD from Columbia College of Physicians and Surgeons and her PhD from New York University School of Medicine. She's an expert in more than 150 primary immune deficiency diseases. Dr. Cunningham-Rundles' research interests are the mechanism and treatment of human immunodeficiency diseases. Her contributions to the field have been widely recognized, and she's received countless awards for her work. Dr. Yaron is the chief of the Hilda and J. Lester Gavrilo Division of Endocrinology, Diabetes, and Bone Disease at Mount Sinai Medical Center in New York. Dr. Tomer received his medical degree from the Sackler School of Medicine in Tel Aviv in Israel and trained in internal medicine in Sheba Medical Center in Israel. He continued his training in internal medicine and endocrinology at the Mount Sinai Medical Center in New York. Dr. Tomer's research focuses on the immunogenetic, epigenetic, and environmental mechanisms causing thyroid autoimmunity and type 1 diabetes. Dr. Tomer serves on the editorial board of Thyroid, and he's a member of the American Society for Clinical Investigation and a fellow of the American College of Physicians, as well as the American College of Endocrinology and a member of the Inter-Urban Clinical Club. Dr. Ioannis Tassiulas recently joined the faculty at the Icahn School of Medicine at Mount Sinai and is an associate professor of medicine in the Division of Rheumatology. He completed his residency in internal medicine at the Albert Einstein Medical Center in Philadelphia and his rheumatology fellowship at the Hospital for Special Surgery. His research interests are in cytokine interactions in the JAK-STAT signaling pathway and regulation of the FCGR signaling and function by the TPL12 kinase. He's an accomplished researcher, mentor, and clinical educator. Please join me in welcoming Dr. Charlotte Cunningham-Rundles, Dr. Yaron, and Dr. Ioannis Tassiulas.
So you don't need to know what that picture is. It's from our roof, which is very nice. So cells of the immune system. So all of us are going to be talking about immune molecules one way or the other. And you probably don't realize allergy immunology does a lot of allergy, yes, but we also look at immunological molecules, immunologic mechanisms. And this is kind of our platform. This is the cast of characters. In terms of which of those cells have immune defects, basically any and all of them can or will do. And as the introduction was saying, there's really a tremendous number of primary immune deficiency diseases now that we recognize. I know that everybody in this room thinks, I'm an internist, why do I need to know this? And I have to tell you that pretty much everything that ever appeared in childhood or infant life in terms of an immune molecule is also present in internal medicine. And you will see them and you will know a little bit more about it as I go forth. All of these immune deficiency diseases are present pretty much in every population. We kind of divide them up because, after all, you can't think about every single disease when you meet a patient. You have to sort of hone in and say, well, which one am I going to concentrate on? Is it going to be an antibody problem or a T-cell problem? Is this a granulocyte problem? And if you go back, then the biggest kind of number of all the diseases that come our way in terms of in medicine are things that go wrong with B cells. Probably because these are the ones that allow you to thrive and to reach adult life, and yet maybe you don't make antibodies to everything. In terms of our own medical center here, we see a great number of people with very late B-cell defects of all sorts. These right here tend to be slightly more on the pediatric zone, but as I say in a minute, all of them are actually present in adult medicine, and we have patients who are 40, 50, or 60 years old who have diseases of XLA, which means no B cells, but of course we keep them alive with gamma globulin. In terms of sort of the clinical big picture here, in terms of not only just my work, obviously I'm very interested in B-cell defects, and that's where I've spent a lot of time. Our biggest disease here at Mount Sinai is common variable immune deficiency. But when I look at this slide, I'm also thinking, but if you open Cecil's textbook, if you open Harrison's textbook, you will find exactly the same figure, which is that if you're going to concentrate on immune defects of adults, you're going to spend a little bit more time thinking about what's going on with the B cells. So all of these, about 75% of the pie here, are things that go wrong with how do B cells work. In terms of the levels of gamma globulin, of course, are low. It's hypogammaglobulinemia. The IgG, the A, and the M are low, and there's not much antibody production. And these are the complications. What would you think? Well, infections is number one. But there's something funny cropping in right here, and that is number two is actually autoimmunity. And that's where it's kind of a boondoggle, isn't it? You think, you're not making antibody, and yet you can have autoimmunity. So there's something innately kind of contradictory about that notion, isn't it? And that's the honest truth, is that this is the way that many of our patients get referred, is they actually have a huge amount of autoimmunity. Biggest one for us is ITP. I think in the ASH guidelines this year or last year was inserted the idea if you see a new patient who has ITP, part of the workup is to get quantitative immunoglobulins. That's because a certain % of those patients actually have that as their root cause. Number two here is Evans syndrome, which is ITP and hemolytic anemia. And number three is autoimmune hemolytic anemia. And again, it seems very unfair, doesn't it? You have somebody who can't bother to make a gamma globulin to Pneumovax. On the other hand, they can get Coombs positive hemolytic anemia, such as this guy right here. So he's 46 years old. He lives in Westchester County. He was referred to me by Hematology. He had a tremendous amount of difficulty with the hemolytic anemia 3 times, quite bad. Ended up with a splenectomy and clots and such. Then had a pneumonia, collapsed lung, empyema, strep pneumoniae. Here's his empyema here. And this is his gamma globulin compartment. And if you look at that, I know you're probably don't look at these all the time, but it doesn't take too much thinking about that to realize that if you add them all up, it's less than 100, and that is basically gamma globulinemia. How can you be gamma globulinic and be 46 years old? So this is kind of the quandary here. So why does autoimmunity develop in the immune deficiency diseases? I'm just going to tell you just a couple of stories about that to try to explain how these two sort of innately oppositional ideas can come together. First of all, I look at immune activation as an immune deficiency. Essentially, anyone who has an immune deficiency also has overactivation, because the immune system is always trying to figure out what to do next. And in doing so, generally will make a few wrong decisions, and the wrong decisions oftentimes involve autoimmunity. These, of course, are kind of the major components. You have the T cells, and you have the B cells over here. Then you have toll receptors that are stimulating interferon from dendritic cells. And you have another set of dendritic cells over here, which are secreting things that incite B cells and tell them, go forth and do what you would like to do. So these molecules, of course, are very high in lupus. So all of these things give you an opportunity, if you have a mutation, to think of what the system might do about that. And another thing to sort of take into your thinking processes is in the bone marrow, now, everyone here, your B cells in your bone marrow are innately autoimmune. They're innately polyspecific, because that's the price that you pay for being able to respond to any antigen. Come what may, you can respond to any antigen. But how do you think that happens? Because you have way too many B cell receptors that are in fact either polyspecific or even autoreactive. And all of them are weeded out over time by what's called central tolerance or peripheral tolerance. So your immune system is using various techniques in order to eliminate autoimmunity at every turn, starting in the thymus over here. And there's many ways of looking at that. And one of them to look at that, and we've done probably around, 15 or 20 papers on this topic alone, which is that the molecules that we see in the autoimmune world are exactly the same molecules that prevent autoimmunity from occurring in everyone else. So right in here, for example, XLA, X-linked agammaglobulinemia, the B cells never develop, but in fact, if you look at them closely, they're all autoimmune in any case. And so this kind of like, you have to remember that that's sort of the way that we're all born. We're born with an incredible capacity, but the price of the capacity is going to be autoreactivity. Now, there's another sort of side of the coin, too, is that there is such a thing as gain-of-function genes. In other words, they don't just lose function, they can gain function. And when they gain function, exactly the same molecule can cause an entirely different phenotype. I'm just going to give you an example of that. Do you remember what Job syndrome is? Everyone kind of remembers some places in the recesses of your memory, Job syndrome. hyper IgE, Buckley-Job syndrome. You have tremendous amount of IgE, you have eczema and boils, huge amount of IgE in the blood, and the lungs get all sorts of infections and pseudocysts. The brain has funny little marks as well. The teeth have different eruption. There's sort of a candidiasis. This is considered a loss of function. So Job syndrome means that you have a mutation in a gene, which is called STAT3. And these are many of the mutations that occur in those patients, pretty much scattered all over the molecule. Everyone, of course, was tremendously excited when this developed because STAT3, who knew what STAT3 even quite did? It's downstream of a number of cytokine receptors, but how could that ever be so important for fungus, for teeth, for brain, for lungs, for staph? It's still a bit of a mystery how all of this develops. So this is kind of the loss of function side of things. But last year, 2004, in Nature Genetics came out of an article that says yes, but there's another side of the coin here, which is that you can have severe autoimmunity due to gain of function of the same gene exactly. Early onset autoimmunity, ITP, hemolytic anemia, organ infiltration of lymphocytes, insulin-dependent diabetes, pretty bad IBD, and loss of normal B-cell immunity. So the patients that we see who come in the door with this have generally become extremely hypogammaglobulinemic, but on the other side of the coin, they have a very bad-looking X-ray. They tell me they've had hemolytic anemia or ITP in the past. The spleen is big. Maybe they have ITP as some degree of enteropathy as well. So it's kind of a surprise, I think, that gain-of-function mutations, which generally, by the way, are even sporadic. You don't get a family history in, I would say, 70% of the patients for anything in their family, even though they may have inherited the gene from their mother or their father. So there's a lot to unravel about how gain-of-function gets permission to misbehave in certain people, but not others. So STAT3 loss of function is on the one side, and that's the hyper IgE syndrome. But gain of function is something quite different. There's something else on here that's kind of troubling, which is the other side of the gain of function mutations is this is present in LGL, large granular lymphocytic leukemia. So somehow STAT3 and the gain of function modality even tends towards cancer. Sort of continuous stimulation is really not a good thing in immunity. So these are article that just came out. Stephanie Vogel put together all the mutations in STAT3, and I have to remember the key here. The gain of function are the black ones, the loss of functions are the green ones, and the blue ones are the ones that go to cancer. So I don't know about you, but I find that very hard to dissect that figure because it's not as if each domain has a specific reason. Would have made a lot kind of more sense to me, and probably to everyone as well, but it doesn't. Scattered throughout this gene are a lot of reasons to think that there's sort of functional aspects we don't completely understand. But it sort of tells you that, immune deficiency can go either way, and it kind of makes that point very clearly. And this is going to be true of pretty much all of the cytokine receptors, whether it's a STAT1 gain of function or something that's just too much of something, but technically it's still due to an immune deficiency. So basically we claim all the immune deficiencies in our division. So even if they present in another area. So this is just one more example here. It kind of hooks to what Dr. Tomar might be wanting to talk about, which is this is a 19-year-old girl with progressive lung disease and enteropathy. Lymphocytic infiltrates in her lungs. No one's lungs are supposed to look like that. She had a biopsy, a lot of infiltrates, reactive germinal centers, non-caseating granuloma here. She's a little bit immune deficient. Her IgG and A and M aren't particularly normal. We fretted about her a lot because she has a tremendous amount of lung disease, so much so that you are thinking, this girl, is not going to have a normal lifespan no matter what. And on whole exome sequencing, she has a mutation in a gene called CTLA4. The CTLA4 is a very interesting molecule because it's in charge of slowing T cells down and putting a brake on. So she has a mutation in the brake here. And again, it's immune deficiency, but it's an immune defect of the brake. So she appears in one camp, but in fact, it's actually due to an immune excess. So this is a patient, if you know what a abatacept is a patient who may in fact thrive under that treatment because that's a way of giving her back that CTLA4 attached to a pharmaceutical compound. So to me, I think autoimmunity and immune deficiency are basically two sides of the same coin. It really is the same thing, it's the same gene, but in fact it has a controlling activity, and it either can be a deficiency or an excess. And that's kind of my bullet there. Your bullet.
Okay, good morning, everyone. Actually, that was a very nice introduction for me, since it was highlighted the existence of autoimmunity as a result of monogenic mutations. What I would like to talk about today in the next 15 slides, hopefully the next 15, 20 minutes, is to go over a polygenic systemic autoimmune disease, which is systemic lupus erythematosus, and give you an update of what is new as far as genetics, pathogenesis, and potential therapeutics in lupus. The current model of autoimmune rheumatic disease development, and that applies mainly in lupus and rheumatoid arthritis, where we have most of the studies, is that there is an interplay between genetic factors, environmental factors, and stochastic events that lead to the development of what is called benign autoimmunity. And we can recognize benign autoimmunity in the clinic mainly by detecting autoantibodies in the serum. Of course, there is evidence in experimental animals that the benign autoimmunity concept includes also the expansion of autoreactive lymphocytes, T and B cells. After the development of the benign autoimmunity, there is another period where either the same or different genetic or environmental factors cause further changes both in the autoantibody specificity by increasing the affinity of the produced autoantibodies, changes in the immunoglobulin subclasses, or changes in the glycosylation of the IgG molecules, clonal expansions of autoreactive B and T cells, and changes in other effector tissue cells, like differentiation and increased activation of osteoclasts and fibroblasts. Still, we're in the benign autoimmunity preclinical state of the disease. Not all of these patients, they will develop clinical disease, but some of them, they will go on to develop clinical symptoms, and they will get a diagnosis of lupus, rheumatoid arthritis, or any other systemic autoimmune rheumatic disease. So from this model, we can think that we have a window of opportunity trying to prevent by treating and suppressing early this autoimmune reaction. We have been very successful to do that in mice when we know when the disease starts, but we haven't figured out yet how to select people that they will develop clinical disease after the development of preclinical autoimmunity. So what are some of the common environmental risk factors for the development of systemic lupus erythematosus? Basically, these data come from large epidemiological studies that have been done all over the world, and factors like smoking, exposure to UV light, certain viruses. The best example here is the infection with Epstein-Barr virus, decreased levels of vitamin D, and of course, exposure to certain drugs, the most common of them that we know for a long time, procainamide and hydralazine, that can induce not only the presence of autoantibodies, but in some cases, clinical lupus. And lately, the last 10 years, with the use of TNF inhibitors for the treatment of different inflammatory conditions, rheumatoid arthritis, IBD, etc., TNF inhibitors, they are now classified into the drug-induced lupus. So there is certain environmental risk factors for the development of lupus. Keep in mind that some of these factors have been associated for the development of other autoimmune diseases, like smoking in rheumatoid arthritis. And I think there is more and better data linking smoking in the pathogenesis of rheumatoid arthritis than lupus. Now, the last 10, 15 years, there is many case control genome-wide association studies done in populations that they have lupus compared to normal controls. And what we learned from all these studies that they have done in different parts of the world is that there is more than 40 polymorphisms in different genes and certain chromosomal loci that are associated with increased risk of developing lupus. And as you can see here, you can classify and categorize these genes into different pathogenic pathways or mechanisms that can help us think the pathogenesis of lupus. You can see here that there is certain polymorphisms in genes that regulate dendritic cell function and type 1 interferon production, other genes that regulate T and B cell function, genes that regulate immune complex or apoptotic cell clearance, genes that regulate cell cycle, apoptosis, and cellular metabolism, and transcriptional regulators. Finally, there is another set of genes that they cannot be classified in any of these categories of the immune-related genes. Some of them, they have to do with tissue damage and severity of damage. So what are some of the lessons from this genome-wide association study? First of all, with the exception of MHC class II, which was known that it's a gene associated with increased risk of getting lupus if you have a certain genotype, and has an odds ratio of about 4 to 5 for developing lupus, the genetic risk loci that have been identified by these studies involve a number of relatively common alleles, each of which confers only a modest risk. So the odds ratios are between 1.5 and 2. Some of these polymorphisms are shared between autoimmune diseases. So there is certain susceptibility genes that confer increased risk not only for developing lupus, but for developing other autoimmune diseases like rheumatoid arthritis, multiple sclerosis, etc. And these are data coming from genome-wide association studies. And finally, and I think most importantly, they provide insight because they identified certain functional pathways that are involved in disease pathogenesis. So this is a rather complicated slide, but in the case of lupus, there are 3 main pathogenic pathways that have been identified based on the genes that came out from these GWAS studies. One set of genes is involved in clearance function, clearance of apoptotic or dead cells, or clearance of immune complexes. Genes that are involved in innate immune activation, type 1 interferon production. Genes that are involved in antigen recognition receptor signaling in the cells of the adaptive immunity, T and B cells. And finally, there is another set of genes that is involved in tissue damage. This concept of preclinical rheumatic disease or preclinical autoimmunity is defined as the presence of abnormalities, as we said, in immune function and immune responses in the absence of clinically manifest tissue injury. And there is good data coming from the United States, the Netherlands, and the Scandinavian countries, where they have big repositories of serum of people, and they are able to go back and study the presence of autoantibodies and other cytokines and chemokines in patients that they develop a disease years before they develop the disease. So there is this famous study done and published 10 years ago here that used this serum repository from the US Army, where they took 130 patients that they diagnosed with lupus, a same group of controls, and they studied the presence of autoantibodies years before they develop clinical disease. So some of the results of these studies are the following. First of all, positivity for ANA in a titer higher than 1 to 120 was detected in 78% of patients that developed lupus, a mean of 3 years before the clinical diagnosis of the disease. Certain autoantibodies were detected earlier before onset of SLE than other autoantibody specificities, and these specificities include ANA, antiphospholipid antibodies, and anti-Rho and La antibodies. And finally, the more specific, and some of them potentially pathogenic autoantibodies, like the anti-double-stranded DNA, SM, and RNP, the positivity for these specificities increased considerably in the year immediately preceding the clinical diagnosis. And you can see here in this graph, this is the time of diagnosis or the first clinical manifestation of lupus. And you can see here the presence of autoantibodies in the serum of these patients many years before the development of clinical disease. Now, having known all these disease susceptibility genes from the case control studies, we learned a lot about pathogenesis. So we identified some genes that are involved in the pathogenesis of lupus, and we took these genes and we went back either in animal models or in vitro studies from human tissues, and we tried to identify what is the role of these genes in the pathogenesis of lupus. And as we said, there were mainly 3 different pathways, pathogenetic pathways, that were identified. One very important pathway is the activation of the innate immune system by nucleic acids. And we now think that this might be maybe the main initial trigger for development of autoimmunity and finally lupus. And in this pathway, there is a very important role of neutrophils and a specific death that neutrophils undergo in the presence of immune complexes, high levels of interferon, which is called netosis. So these neutrophils, they die by excreting what they called neutrophil extracellular traps. And in this mess of histones, other proteins, and DNA, there is presence of autoantigens, which is the DNA in lupus. And we think this is one of the main antigenic sources in lupus. We defined the role of the major type 1 interferon producers in lupus, which is the plasmacytoid dendritic cells, and also defined the role, the very important role of type 1 interferons as a major driver of autoimmunity in lupus. So schematically here, you can see that apoptotic cells or apoptotic particles or immune complexes that contain nucleic acids can get access to dendritic cells, plasmacytoid or myeloid dendritic cells, and can stimulate, can be recognized by toll-like receptors that recognize nucleic acids and stimulate the production of type 1 interferon. In lupus, increased production of type 1 interferon was known from the late 70s, but nobody really knew what are the cells that produce that and what exactly is the role of type 1 interferon. Now, in the cytoplasm of all cells, there is other nucleic acid recognition receptors that can recognize and can be activated by nucleic acids coming from bacteria and viruses. Again, the activation of these receptors leads to increased production of type 1 interferon. And this might be the reason, as we know, that bacterial or viral infections are among the most common triggers of lupus flares. What are some of the known abnormalities in the cells of the adaptive immune system in lupus? The best well-characterized T-cell abnormalities include decreased number of regulatory or suppressor T-cells, increased number of this particular subtype of T-cells, which are called T follicular helper cells, and these are cells that specifically help B cells in the germinal centers for the production of high-affinity autoantibodies, and also decreased production of interleukin-2 and increased production of IL-17 and IL-21. Now, decreased production of interleukin-2 by T cells in lupus was known for more than 30 years. Nobody really knew exactly what that meant and why that happened. It was common to say that T cells in lupus are exhausted, so they don't produce a lot of IL-2. But it turns out that they are not exhausted, but there are some changes in the signaling downstream of the T cell receptor that shifts the production of cytokines from IL-2 to other pro-inflammatory cytokines like IL-17 and IL-21. IL-17 is important, mediating inflammation in target organ and tissues, and IL-21 is one of the main cytokines that drives the differentiation of follicular helper T cells. B lymphocytes abnormalities in SLE. Of course, the major hallmark in HLE is the production of autoantibodies, and there are many different defects in checkpoints of B cell development and maturation that have been described in lupus. And also there are many different receptor molecules and cytokines that drive the production and the increased differentiation of auto-reactive B cells in lupus. Finally, what mediates damage in lupus? Because we said you can develop autoimmunity, but this does not translate necessarily to clinical disease and damage. And I'm going to spend 2 minutes telling you about immune and non-immune mechanisms that mediate damage in lupus nephritis. And as you know, kidney is a major target organ in lupus. We know that the first event in lupus nephritis is the deposition or the in situ formation of immune complexes that can be deposited either in the mesangium, in the subendothelial or subepithelial part of the basement membrane. But we know that just the deposition of immune complexes does not cause inflammation or damage. And we know that from animal studies. Now, these deposited immune complexes can activate endothelial cells that can start produce chemokines and cytokines that they will attract other immune cells, and they will perpetuate the inflammation. In human and mouse lupus, many different inflammatory mediators have been detected in increased level in lupus, including chemokines like CCL2 and cytokines like TNF, interleukin-17, etc., and also cellular infiltrates composed of dendritic cells, neutrophils, macrophages, and clonally expanded T and B cells. Now, it's becoming obvious that there is other non-immune mechanisms that play a role in damage in lupus nephritis, and maybe this is the part that we don't target with our immunosuppressive treatments these days. And yes, we're doing much better in treating patients with lupus, but definitely we have a long way to go as far as treatment. We know that endothelial cells, podocytes, and mesangial cells communicate and talk to each other and secrete certain growth factors and survival factors. So there is many different abnormalities described in the secretion and production of these factors in lupus nephritis, some of them specific for lupus nephritis, some other that they are common with different glomerulonephritides. And the same is going on in the interstitium, like the glomerulus, and much more, there is the development of fibrosis. And fibrosis, it's usually an end stage in lupus nephritis, and it's something that we don't have a good therapeutic mean to target. So having said all that, what are the implications for treatment, having all this knowledge? First of all, targeting the immune system, the systemic autoimmunity, and thinking the preclinical autoimmunity, it seems that there is a window of opportunity for primary or secondary prevention of damage in autoimmune diseases. We've been successful in doing that in animals because we know exactly when the autoimmune reaction starts, but it will be much more challenging to do that in humans. Possible targets of the immune system include cytokines, and you can pick and choose your best cytokine or your favorite cytokine, and there is different antibodies and molecules targeting all these cytokines in clinical trials. Signaling molecules, kinases that mediate signals downstream of different receptors, co-stimulatory molecules, plasma cell depletion by the use of proteasome inhibitors, and these are in clinical trials. And also, lately, there is some trials that they try to increase the number of regulatory or suppressor T cells. Of course, by targeting only the autoimmunity, we don't address the non-immune cells that mediate damage and injury in lupus. So targeting endothelial cell dysfunction, there is some evidence that CellCept or MMF improves the function of endothelial cells. ACE inhibitors and endothelin antagonists are other options. And targeting fibrosis, I think there is a couple of antibodies targeting TGF-beta, and there is one successful trial in systemic sclerosis. So that's something that's coming up for treating lupus nephritis. And finally, 1 month ago, there were, in the American College of Rheumatology meeting, there was presentation of a monoclonal antibody blocking the type 1 interferon receptor in lupus, and that was a positive trial. We haven't had too many positive trials of biologic in lupus, so that's very exciting. It's in phase 3 trials. And also another open label trial coming from China, but there was another small open label trial from Germany by using low-dose IL-2, try to increase differentiation and number of regulatory T cells, and that has been done successfully in lupus. Keep in mind that this is an open trial. And I think I'll stop here. Thank you.
So I would like to continue the theme of genetic and mechanistic studies leading us to new treatments. I have nothing to disclose. This is a review that was published by Von Herath and Dirk Hohmann here from Sinai about the burden of autoimmunity. And as you can see, autoimmune diseases in general are very common. The top two most common autoimmune diseases are autoimmune thyroid diseases, Graves and Hashimoto's. It is estimated that there is about 10 million patients in the US with Hashimoto's and over 2.5 million with Graves' disease. So these are definitely common diseases. The general scheme of the mechanisms, as was described just for lupus, is that there is some genetic susceptibility. The individual inherits a combination of susceptibility genes, and with interaction with environmental triggers, such as infection in the case of thyroid disease, iodine, pregnancy, there is an epigenetic interaction in which the environment turns on or off certain genes that leads to infiltration of the thyroid with T cells targeting the thyroid. If these T cells will undergo Th1 differentiation, and they will lead to apoptosis of thyroid cells and hypothyroidism or Hashimoto's. If they will undergo Th2 differentiation, they'll lead to the production of TSH receptor stimulating antibodies by B cells and hyperthyroidism or Graves' disease. And our focus, or one of areas that we've been focusing on in the past 20 years, is finding genes as clues to the mechanisms. Now, the genetic studies in thyroid autoimmunity and in most complex diseases underwent several phases. The first phase was the candidate gene phase. Genes that looked like they may be important, such as HLA, were tested whether they were associated with disease. The second phase, this is actually the phase where I entered the field, was taking families and looking for linkage between markers in the families and the disease that you're studying. Third phase that got a lot of publicity is the GWAS phase, taking thousands of patients and controls and screening the whole genome with densely spaced markers. The fourth phase, which is now in full swing, is sequencing the whole human genome. As Charlotte just mentioned, for immune deficiency, it can lead to identification of rare variants that will not show in the GWAS that only looks at common variants. And the fifth phase, going back to the mechanism, is to see how these interact epigenetically. I won't have time to talk about all the phases, but I'll start with just a few words about GWAS. A lot has been said about GWASs with a lot of criticism, especially the low odds ratios that we find. And some people always ask us, especially patients, if there is a genetic association, can this tell me if my children will develop disease? So I wanted to put this graph here that shows, actually as was just discussed, most of the odds ratios in GWAS are even between 1.5 and 2. Odds ratio of 3 is considered terrific. So if you have an odds ratio of 3 and you want to have, this is the true positive on the y-axis and the false positive, 80% true positives, you'll have 60% false positives. And for a marker to be really clinically useful, you need odds ratios of over 100 so that the true positives will be over 90% and the false positives less than 10%. So in all reality, GWAS studies will never lead to a marker that will tell us whether the patient can develop disease, but they yield a lot of useful information about mechanisms. The high throughput sequencing is now in full swing. And this is mostly due to technological advances. So this is how the human genome was sequenced in 2000. It took 14 years and cost $2.7 billion. And in 2015, we have a desktop sequencer that in 24 hours can sequence the whole human genome for $1,000. And this is now not just in research, but this is actually going into clinical diagnosis. You see this paper about the FDA authorizing the first sequencer for clinical use. It's used mostly now for in pediatrics, children that are born with some syndromes that are unclear. Instead of doing karyotype and so forth, you can just sequence the whole genome and find the mutation. But it will be probably expanded to cancer, immune deficiencies, and other areas. And this is expanding so much that it is coming to a neighborhood near you. This is not a montage. This is a paper. This is from the Wall Street Journal. These are going in neighborhoods. So if you go to Best Buy with your child and you want to check if he or she is really your child, you can give a little bit of a buccal smear and you'll get an answer. And if you don't like the answer, I guess the lawyer's truck is behind. So as was said before, all the genetic studies didn't just lead to some random list of genes, but these genes actually show themes. In the case of lupus, you've seen the theme of clearance of DNA and antigen presentation. In the case of autoimmune thyroid disease and type 1 diabetes, what we're seeing is that most of the genes that were found to be associated with disease are involved in antigen presentation. This is the HLA class 2 that's presenting a peptide antigen to the T cell receptor and the co-stimulatory molecule CD40, CTLA4, CD28. And as you can see, most of the genes that were found to be associated are participating in antigen presentation, the HLA itself, the antigen, CD40, CTLA4. We've focused my lab for the past 15 years on the CD40 gene that we found through whole genome linkage studies. And through sequencing, we identified a SNP in the Cossack sequence, which is important for initiation of translation. A CT SNP, or the CC genotype, was associated with Graves' disease. And later, CD40, given that it is an immune gene involved in antigen presentation, was reported to be associated with a whole list of other autoimmune diseases. So the question is not now that we have this SNP, as I mentioned, I'm not going to use it to predict who will develop Graves' disease. That's impossible because 70% of normal individuals still have the susceptible SNP. But can it tell us about the mechanisms of disease, and can these mechanisms then lead to translation? So as I mentioned, this SNP is in a region of the gene that is important for the RNA to bind to ribosomes and initiate translation. And the question was, could this affect the translation and expression of the gene? And this is just one of many experiments that we did that show that if you transfect cells with a susceptible allele, you get higher levels of CD40 expression than with the protective allele. So the question is, we know that CD40 is expressed on B cells, so if this allele makes you make more CD40, it will probably augment B cell function. But CD40 was also known for many years before we started our studies to be expressed on thyroid cells. And we were wondering whether its expression on the thyroid targets the immune response to the thyroid in Graves' disease. So we created mice in which we put a transgene of the CD40 in the thyroid. So these mice overexpress, this is the brown here, CD40 in the thyroid. These are the transgenic mice and the wild-type mice. And then we induced Graves' disease in the mice. In mice, unlike in humans, if you want to know if they have Graves' disease, you can just take out their thyroid. So this is a normal mouse, and this is the normal thyroid with a single cuboidal epithelium, and this is a mouse with Graves' disease. You see the goiter, you see the proliferation of thyroid cells, and if you take blood from them, they have TSH receptor antibodies and high levels of T4. And when you compare the transgenic mice that had excess CD40 expressed, overexpression of CD40 in the thyroid compared to the wild type, they had more severe disease. They had high levels of antibodies and significantly higher levels of T4, suggesting that the CD40 overexpression in the thyroid is responsible for augmenting and accelerating the disease. But then the question is, okay, CD40 in the thyroid, but what does it do there? So we wanted to check the signaling pathway of CD40. CD40 is a member of the TNF receptor family, and without going through all the steps, what the main point here is that there is a canonical pathway that leads to this expression of these cytokines, and a non-canonical pathway which leads to BAFF and BLyS, which is a major B cell regulator. So we tested the signaling pathways of CD40 in thyroid cells, and both were activated when you activated CD40 on thyroid cells. Both the non-canonical, as shown here by BAFF, increased expression when you stimulate CD40 on thyroid cells. This is not on B cells, and interleukin-6. So The translational implications are that we can target these two. If we believe that CD40 gives you the genetic susceptibility, makes the thyroid express more CD40, and this will lead to excess BAFF and interleukin-6, which are involved with disease, what happens if we target IL-6 and BAFF? So this is a study that was done in 2006 by Gilbert et al. In mice, again, with the mouse model of Graves' disease. And when you block BAFF with two different methods, the disease was suppressed. And we tested the effects of blocking IL-6. And again, these are the mice that got anti-IL-6. And you can see that the T4 levels are all normal. This is the cutoff, while the mice that didn't get IL-6 were thyrotoxic, and the same for the antibodies. And is this actually in clinical trials? So there are antibodies right now in FDA approved for other indications. Anti-IL-6 is called tocilizumab, and it is approved for rheumatoid arthritis. And there is now a study in Spain that will finish enrollment in 2016. So we're eagerly waiting to see the results, in which IL-6 is given for Graves' ophthalmopathy. Anti-BAFF has not been studied yet in Graves' disease, but hopefully this will be picked on and studied. I do want to make here a side comment, since people mention monoclonal antibodies all the time, and the nomenclature is very confusing. So as a public service announcement, how do you look at all these names? So this is the murine antibody. All blue is murine and brown is human. We don't have any ones in clinical use, so I don't have an example. But what you look is before the MAB, monoclonal antibody, the letters tell you what kind of antibody it is. So abciximab with XI is chimeric. It has only the variable region from the mouse. And if you have ZU, like tocilizumab, it is a humanized antibody. So the hypervariable region is still from the mouse. Everything else is from human. And if it's a human antibody, then you'll have a U here, like in adalimumab and belimumab. So now you know how to read this confusing nomenclature. I want to use the last few minutes to talk about another gene of interest. Everybody knows that HLA is associated with autoimmunity, and HLA-DR3 has been known for decades to be associated with thyroid autoimmunity as well as other autoimmune diseases, specifically HLA-DR3. But HLA-DR3 is not just one gene. It has over 40 different variants. So a few years ago, we wanted to see if there is a specific variant that's associated with thyroid autoimmunity. And what we found is that indeed, if you have an arginine at position 74 of the beta chain, so the HLA has an alpha chain, which is constant, and a beta chain, which is variable and determines the specificity, whether it's DR1, DR2, DR3. And if you have arginine at position 74 of the variable beta chain, you are susceptible to Graves' and Hashimoto's, and glutamine was resistant. And these are actually different amino acids. In this figure, in this crystal structure, blue is positive charge, red is negative charge, and white is hydrophobic or nonpolar. And arginine is a large positively charged amino acid, while glutamine is smaller and neutral or negatively charged, partially negatively charged. So we hypothesized that this change will change the peptide presentation. As I mentioned, antigen presentation is at the key of the genetic susceptibility. So could this pocket accommodate only specific peptides and not others? And indeed, this is what we found. Again, summarizing one slide, 5 years of work. We found 4 thyroglobulin peptides that fit specifically in the arginine pocket and not in the glutamine pocket. What was common to them is that they all had a negatively charged amino acid here that interacted with the arginine to sit comfortably in the pocket, while those that didn't have it were being pushed out of the pocket. So this is all nice, but can it have translational implication? And to us, the translational implications were, if this is really the key, this HLA enables you to present thyroglobulin peptides that will trigger disease, can we block it? So the idea is, here is the antigen presenting cell. It's presenting the thyroglobulin peptide to the T cell. Could we block this interaction? And to test whether different molecules could block this interaction, we developed this ELISA assay where we took recombinant HLA-DR3, we incubated with the peptide and for 24 hours, and then we add them to plates that are coated with antibody that binds the HLA, and we wash it out. The peptide is labeled, so if the complex bound, if the peptide bind to the HLA and the complex bound, you'll see the signal. If this molecule that we're screening for, that we think is blocking the peptide from binding to HLA, blocks the pocket, then less peptide will bind and you'll have less signal. So the decrease in the signal will tell you the activity or the inhibition of this molecule of peptide binding. And we screened virtually over 100,000 molecules and came with 57 candidates that we tested with this ELISA. And to show these are 3 candidates that look promising, that have over 50% inhibition. And in preliminary results that we did in mice, we saw that testing these small molecules in the mice actually shows inhibition of T-cell proliferation in mice, both when we take their T-cells and do it ex vivo or when you give the small molecule in vivo to the mice. So the hope is that maybe one day this will be used in humans, but the point is that this was all started by just doing mechanistic studies. In fact, we're very hopeful because this specific molecule is on the FDA list, so it's not just a random molecule, it's a molecule that is actually being used in humans for other indications, so if it is confirmed, maybe it could be repurposed for thyroid autoimmunity. So in summary, most autoimmune thyroid disease genes participate in antigen presentation. As I mentioned, CD40 is a major gene for thyroid autoimmunity. Blocking IL-6 and BAFF may be a new approach to therapy, and there is at least one clinical trial right now. The R gene in 74 is a major allele for autoimmune thyroid disease, and hopefully in the future, maybe we'll be able to block antigen presentation by this R gene and prevent disease. So these studies are done with a lot of collaborators from here and from other institutions, and this is a picture of the members of my lab. Thank you.
We have time for a few questions. What other questions? Yeah, thank you for staying with the leopard in the laboratory. It's so frustrating, but you guys are spending a career doing such good work. My question is about the process of aging, not AGED, AGED, but the process of aging and autoimmunity. And clearly, some diseases in very young people, like lupus, some in middle-aged people, autoimmune disease like diabetes type 1, and some in older people, whether it's rheumatoid or some wild ideas that Alzheimer's may be part of. But would you comment on whether you or your colleagues have been working at all on the biology of normal aging and its impact on autoimmunity?
Well, the first, almost the first study that I did as a resident many, many years ago was actually about that, because in aging you have more autoantibodies. So as you age, you have, if you measure, and we all know that our globulin antibodies at age 20, you may have it in 1% of the population, can go up to, and rheumatoid factor and so forth. So the question is, we know that autoimmune diseases are actually diseases more of middle age or women of reproductive age, at least those that are more common in women. So how come as you age, you have more and more autoantibodies? Are they pathogenic? And to summarize studies, not just that I did, that little study, but many people, those antibodies are probably not pathogenic. So they are there. There was even a study out of Pisa of one of our former fellows in endocrinology that suggested that they're even protective and make you age longer, because when they looked at people 100 years and over, they even had higher levels of autoantibodies. I'm not sure that they're protective, but they're definitely not harmful. So it seems as if the accumulation of antibodies that you accumulate over as you age does not indicate also propensity to develop disease.
Well, these are two random thoughts anyway. Number one is that people make B cells their entire life. A guy named Max Cooper actually did bone marrow samples from ribs from ORs and did an amazing study that showed that between the ages of fetal life up to the age of 80, you're continuously making new B cells, but you can't get around the fact that figure also shows there's a huge burst in the, say, first 20 years of life. So it could be that what you have is this explosion of B cells early on, and then later you're making fewer and fewer and fewer of them. Some of those are most likely regulatory B cells. So that's one thing that sort of says autoimmunity is skewed to the youth. In fact, your B cells are a hell of a lot more active when you're younger than they are when you're a little bit older. And so maybe that's why certain diseases are a little bit skewed that direction. It could be that the T-cell immunity, though, which we also know changes with time, is a little less able to rein in those abnormal clones as well. So there's two sides of the coin, I think, is part of the same story.
I think also besides the genetic factors, there is some other factors that contribute to that, and I think a good example is hormones and estrogens as drivers, at least in expression of lupus. In women of reproductive age, the ratio between females and males, as we know, is about 9:10 to 1. In the postmenopausal age, this ratio decreases about 3 to 4 to 1. So definitely there is other factors from the environment or other factors that we don't even know that contribute to autoimmunity.
Thank you very much for good talks from everybody. You don't have to convince us that genetics is important, whether innate or something that develops later. You did mention environmental factors, and one that you didn't mention was the microbiome, which has been shown in vitro and in vivo studies to actually have an effect on the immune system, and is even being used via fecal transplants for some people with inflammatory bowel disease. Now, how much this affects some of the other areas you've talked about, that I don't know.
At least if I can speak, for the systemic autoimmune rheumatic disease, I'm not aware of any studies of fecal transplant as a means to treat these conditions. But definitely what we understand and what we know is that the microbiome has an important role in shaping the immune responses. And at least for rheumatoid arthritis, there is good data showing that the autoimmune reaction does not really start in the joints, but most likely starts in the lungs. And there is increasing interest of people finding and connecting environmental factors like smoking with interactions with the microbiome in the lung and other tissues. So I'm pretty sure in the next year we'll have much more information about the microbiome in the systemic autoimmune diseases.
So this is an excellent comment, and obviously if we needed to talk about the environment, not just microbiome, it's another grand rounds. But in type 1 diabetes especially, this is a very hot topic because the frequency of type 1 diabetes has been increasing exponentially since World War II, doubling every 20 years. It's actually almost like an epidemic. And one of the obvious suspects would be antibiotics that were introduced after World War II, changing the microbiome. So there's a whole group. In fact, there was a meeting in Chicago just a few months ago in which all the type 1 experts met to see how they can promote microbiome studies. However, the data is still not definitive. We know that the immune system does circulate through the gut and is partially educated by the gut, but I think except for IBD right now, still the data is not proven, but this is definitely an area that requires further studies.
Well, you can imagine readily if you don't have any IgA, which most of our patients who have common variable or antibody deficiency, they lose IgA. So what does loss of IgA do? And I've actually never seen any terrific studies. I think the problem is you need a tremendous number of people who haven't been on antibiotics, right? And I think that the way to do that study is obviously going to be very expensive, but probably very informative. I know that my colleague at Harvard, Scott Snapper, is studying people who have genetic defects and looking at the microbiome in such patients, but remember how much antibiotics are going to mess that up. If they've been on an antibiotic recently, that's going to be difficult. So we all talk about it, frankly. I think actually getting the data on that for people with immune deficiency is going to be pretty hard. We know it's going to be important, but antibiotics are a problem. Don't you think?